Have you ever wondered why you're a totally unique mix of your parents, not just a carbon copy of one of them? Well, today, we're going to pull back the curtain on the amazing biological process that makes all that variety possible. It's an incredible cellular dance called meiosis.
Okay, let's start at the very beginning. For every single one of us, life started as just one cell. That one cell was created when two super specialized cells, a sperm and an egg, met and fused together.
But hang on, that brings up a really big question, kind of like a math problem. If your mom gives you her entire set of genetic instructions and your dad gives you his entire set, shouldn't you have double the DNA? Why don't we see the instruction manual double in size every single generation?
To figure this puzzle out, we have to look at how our genetic information is actually organized. You see, it's not just one giant, messy string of code. It's packaged up really neatly.
So, what's the magic number? It's 46. Inside almost every single cell in your body, you've got 46 chromosomes. You can think of them as 46 separate chapters in the instruction manual that is you.
And these 46 chapters aren't just random. They're organized into 23 neat pairs. For each pair, one chromosome came from your mom's egg, and the other came from your dad's sperm. And because they carry the same types of genes, we call them a homologous pair.
Okay, so back to our big question. How does life solve that problem of doubling the chromosomes? The answer is this absolutely brilliant and essential process called meiosis.
Now, you've probably heard of mitosis. That's the process your body uses to make new cells for growth and repair. It's basically a cellular copy machine. But to make sex cells, you need a totally different strategy. That's where meiosis comes in. It's a special kind of division where the whole point is to cut that chromosome number exactly in half. And meiosis doesn't do this in just one go. No, it's a two-part performance. Let's check out the first act, meiosis 1. This is where the really dramatic change happens.
A really easy way to remember the phases is the acronym PMAT. That stands for prophase, metaphase, anaphase, and telophase. And in meiosis, the cell goes through that whole sequence not once, but twice.
It all kicks off with prophase 1, and seriously, this is where the magic begins. Those homologous pairs of chromosomes find each other, they get super close, and they actually swap pieces of their DNA. This is called crossing over, and it's like shuffling a deck of genetic cards, creating brand new combinations that have never existed before. It's incredible.
So, after that amazing genetic shuffle, these freshly mixed pairs of chromosomes all line up right down the center of the cell. They're getting ready for the big separation, which happens right now in anaphase 1. This is the critical moment. Instead of splitting individual chromosomes, the cell pulls the entire homologous pairs apart. One whole double-stranded chromosome goes one way, and its partner goes the other. And just like that, the chromosome number is officially cut in half. The first division finishes up in telophase 1. The cell pinches in the middle, and we're left with two brand new cells. And the important thing is, each one now has only 23 chromosomes. The reduction is complete. But hold on, we're not done yet. Those two cells, with their 23 chromosomes, almost immediately start gearing up for round 2. This is meiosis 2. Now, this second division is pretty similar to the first, but with one massive difference. In metaphase 2, the chromosomes line up single file down the middle. See that? They're not in pairs anymore, and that sets up a totally different kind of split.
That single file line is the green light for anaphase 2. The cell finally pulls each of those X-shaped chromosomes apart, separating them into their individual strands, which we call chromatids.
And as the cells divide for the very last time in telophase 2, we see the amazing final result. We started with just one cell with 46 chromosomes, and now we have 4 totally unique sex cells. Each one is carrying just 23 single chromosomes, perfectly ready for fertilization. So, if you're going to remember one thing, it's this. Meiosis is a reduction division. It cuts the chromosome number in half during that first division, and then it separates the individual strands in the second one. It's a perfect system for prepping these cells.
Okay, so we've just walked through the how of meiosis. But what about the why? I mean, why does life go through this complicated two-step process?
Well, the huge payoff is genetic diversity. That crossing over thing we saw back in prophase I, plus the random way the chromosomes get sorted, means that the 4 sex cells at the end are all genetically different from each other. That variety is the fuel for evolution. It's what allows species to adapt and survive in a changing world.
And if you want to see what happens when the very precise rules of meiosis aren't followed, we just have to look at the mule. It's a fascinating case. See, a horse has 64 chromosomes, but a donkey has 62. So when they have a baby, the mule, it ends up with 63.
And 63? Well, that's an odd number. When a mule's cells try to do meiosis, they just hit a wall. There's no way to make 31.5 pairs of chromosomes. The whole process basically fails before it can even get going properly, which means mules can't make their own sex cells. That's why they're sterile.
The sterile mule is just a perfect example of the incredible precision that life demands. Meiosis isn't just some random process. It's a truly elegant dance that has to be performed perfectly every single time. It guarantees the right number of chromosomes get passed on, while also creating the very variety that makes life so rich. And it leaves us with one last question to think about. What other profound things can happen when this fundamental, elegant dance makes a mistake?